Hello everyone, I'm Arun and my research topic is Metabarcoding analysis of microbiomes from sewage treatment plant. The problem of conventional sewage treatment plant is that pathogens are not removed effectively in this flowchart as the water is preliminary, primary and secondary treated and let out into water bodies, and these pathogens have the potential to interact with microbiota from rivers and ocean to create a pathogenic microbiome that is harmful for the environment and especially for the marine environment. So the research question here is, are the pathogens filtered in various stages in sewage treatment plant? To answer that, microbiome was isolated from various stages of sewage treatment, namely the influent, primary treatment, secondary treatment, and the effluent, and was sequenced using CNS sequencing. And those sequences were analyzed using R, and the data from R was inputted into Shiny Migal application that gives graphical representations such as heat maps and taxonomic classification. So what does the heat map show? So the x-axis of the heat map are the samples where A to D was from September, which is warmer, and E to H is from December. And the x-axis has various bacterial sequence reads from the reference, and more the red, more the abundance. So in influent, there were like highly abundant and broad range of bacteria that steadily decreased across the filtration process, and in final effluent it was really low compared to that of the beginning. Whereas in case of December sample, there was a wide range that got reduced until the second filtration, and the final effluent had an increase in abundance as well as in the variation of species. Coming to taxonomy classification of the microbiome, so here the x-axis denotes the sample similar to that of the heat map, the first four being September, the latter four being December. And the y-axis denotes the relative abundance of each genera with respect to the other genus. So in this case, the bigger box of Psychrobacter is more abundant than the smaller boxes of Mycobacterium and Trichococcus. So in this case, overall, Mycobacterium is present throughout all the samples and it's significantly abundant. And one of the pathogens in Mycobacterium is Mycobacterium tuberculosis, and Psychrobacter was present in September influent the highest, and there was traces in others, and that could cause acute gastrointestinal infection. And the others denoted in black are Pseudomonas acrobacter, that's really present small in abundance, whereas Trichococcus, the light green one, is present significantly throughout, and it's a non-pathogenic, and it also has biocontrol properties against fungal pathogens. So what we don't have is the highest confidence on whether a pathogen is present or not. So the species analysis would increase the confidence of pathogens in the sewage. And special thanks for Kirsten Lure for obtaining and sequencing the samples, and thanks to Dr. Chen and Dr. Pagsuyoin.